Well, thank you again for the invitation to come. What I'm going to do in the second part of the seminar this morning is I'm going to talk about two things. I want to talk about some of the research that we've done to understand what is the biological basis for the differences that we see in the fluorescence images and in the fluorescence spectroscopy. And then I want to talk about some new technologies that are in pilot clinical trials where we're trying to image with very high spatial resolution and to develop targeted contrast agents that will allow us to be sensitive to some of the molecular changes that are known to accompany the development of these precancerous lesions. And so, as Anne mentioned, we're interested in looking both at wide field imaging and point spectroscopy. So in the first part of my talk, I'm going to focus on why do we see this decrease in autofluorescence when we look at the wide field imaging, and what is responsible for the changes that we see in the spectroscopy as well. And then in the second part of the talk, I'll focus on some new technologies to do very high spatial resolution optical imaging in vivo that would allow you to approximate the kind of resolution that you have in a biopsy without having to take the biopsy. And then talk about some stains that we're developing that you can apply to tissue in vivo that would allow you to highlight particular molecular changes. So these are just repeating some of the slides that Anne showed where we see this loss of fluorescence associated with lesions and wide field images. And here when we look at the spectroscopy data, this is at 350 nanometer excitation. As we go from normal to mild dysplasia to moderate dysplasia in cancer, again, we see that drop in the fluorescence, and we see a little bit of a shift to longer emission wavelengths. And these data presented here were collected with the shallow channel of that ball lens tipped probe. And when you compare that to what we see in the histology, this is the normal oral mucosa, and this is severe dysplasia. With the shallow channel of the probe, we're probing approximately the top 250 to 350 microns of tissue. And that roughly corresponds to the thickness of the epithelium. And there have been a number of people who've looked at what are the changes in the optical properties, both in the epithelium and in the stroma, that accompany the presence of dysplasia. And they include increased scattering in the epithelium with dysplasia, decreased scattering in the stroma, increased fluorescence in the epithelium, and decreased fluorescence in the stroma. We have just concluded a series of studies to look specifically at what happens in the oral mucosa with the development of dysplasia and cancer. And we took two approaches to understand biologically what's responsible for these differences. In the first approach, what we did is we took biopsies of tissue, and immediately after taking the biopsy, we put it in chilled culture media, and then we sliced it using a special tissue slicer so that the tissue is still alive, and then we're able to image the fluorescence of the tissue using confocal microscopy with very high spatial resolution. And that allows us to look at what are the changes in fluorescence with subcellular resolution in the epithelium and in the stroma. And then once we know what those optical properties are, we can do Monte Carlo simulations of light propagation in tissue using the information about the optical properties that we've measured from our oral cavity specimens to understand where the light is coming from and what's responsible for this drop in fluorescence. But actually, one of the first insights that we gained from this was to better appreciate some of the reasons for the differences in both the imaging results and the spectroscopy results from different sites in the oral cavity. If you look at a biopsy of a keratinized site, and this would include the gingiva and the palate, you can see this very thick layer of keratin and then the epithelial cells underneath it, in contrast to a site that doesn't have any keratin. And what I show you now are fluorescence confocal images of living slices of tissue. The top row shows these images with UV excitation. This is an argon at 351 to 364, and here with 488 nanometer excitation. And we've drawn a white line here to indicate the basement membrane. So above that is epithelium, below that is stroma, and the scale bars here are 200 microns. And what you see in these two keratinized sites, the palate and the gingiva, is strong keratin autofluorescence and actually pretty weak cellular autofluorescence. And you see that also at 488. In contrast, the non-keratinized buccal mucosa, you see a lot of cellular autofluorescence in the epithelium, both at UV and 488 nanometer excitation. And so the patterns of autofluorescence of these two types of tissue look very different. And for the rest of the talk this morning, I'm going to focus in on what happens in these non-keratinized sites, which is what Anne showed you the receiver-operator characteristic curves for. So these tissue slices were obtained from the tongue, and these are from a normal area of the tongue. So here's the H&E, here's the epithelium, and here's the stroma down here. And what you see when you look at the fluorescence with UV excitation is particularly on this basal layer of epithelial cells, you see dark nuclei and bright cytoplasmic autofluorescence. And basically that's the NADH associated with the mitochondria that is responsible for this fluorescence. At 488 excitation, again, you see dark nuclei and mitochondrial fluorescence at this excitation wavelength, which is due to FAD, the other mitochondrial fluorophore. And then you see the very bright autofluorescence associated with collagen crosslinks in the stroma. So let's take a look now at what happens as we begin to look at abnormal tissues. And we found it useful in trying to understand what was happening to divide the tissue into layers. And so I talked about the epithelium and the stroma. But within the epithelium, we observed sometimes a very thin superficial layer of keratin, even in these traditionally non-keratinized sites. And then we had this basal layer where we saw bright UV fluorescence, but dim 488 fluorescence. And we had an intermediate layer of cells where we saw bright 488 fluorescence, but dim UV fluorescence. And then we divided the stroma, for reasons that will become clear in a moment, into two layers. A layer that we called the subepithelial stroma, that's about 125 microns thick, and the deeper stroma. In the normal tissue, those layers look similar. So now, as we begin to look at abnormalities, this is looking at benign inflammation. So this can sometimes give rise to false positives, but it's not of clinical concern. What we find with benign inflammation is that there's a big loss of UV fluorescence in the epithelium, as well as a loss of fluorescence in the subepithelial stroma. And you see that the fluorescence in the stroma is also lost at 488 nanometer excitation. And you begin to see some punctate spots of fluorescence here, which we believe are associated with inflammatory cells that are infiltrating in the stroma. In contrast, with mild dysplasia, what we see is an increase in the amount of UV fluorescence in the epithelium and a decrease in the stromal fluorescence, both at UV and at 488 nanometer excitation. And remember, we're interested in being able to tell apart the severe dysplasia from the normal and the inflammation. With cancer, what you see is a disorganization of the tissue structure. You lose the layered nature of the epithelium and the stroma, and you have nests of tumor cells with intervening fibrous matrix surrounding them. And you can see here regions where we have the fluorescence of the tumor cells and the stromal matrix in between. And so we imaged the fluorescence of slices from about 50 biopsies in this study, and then we divided those biopsies which were normal, inflammation, or dysplasia into these 5 layers, the superficial, intermediate, basal, and the 2 stromal layers. And we looked at what was the average fluorescence from each of those layers, both at UV and 488 nanometer excitation. And so here we've got the superficial, the intermediate, the basal, that subepithelial stroma, and the deep stroma. And I'm showing you here normal tissue without any inflammation, normal tissue with mild or moderate inflammation, normal tissue with severe inflammation, and dysplasia. And in this study, all of our dysplastic samples also had inflammation associated with them. And so remember, we're interested in telling apart normal and inflammation from dysplasia. And so what you see is that in the basal epithelium, the dysplasias have higher fluorescence than the normals or the inflammation. But when we look in that subepithelial stroma, both the dysplasia and the inflammatory specimens are losing fluorescence there compared to the normal tissue. And that was one of the reasons that we were so interested in looking at the probe that had the depth selectivity, because it could help us separate the signal from the basal epithelium and that subepithelial stroma. And this just illustrates with traditional probes where you have fibers that are pointing straight down at the tissue, the region that you're collecting light from is the region where these cones of light from the fibers overlap. And either with the ball lens or using an angled geometry, as other people have done, you can confine the region of overlap more to the epithelium. This is our ball lens probe again, and what we did was we did Monte Carlo modeling to trace the input of light into the tissue, track the propagation of photons through the tissue, and then determine the resulting collected photons with the probe. And to demonstrate, to validate our Monte Carlo simulations, we looked at the intensity of fluorescence collected from a very thin layer of fluorescent material. As that fluorescent material, that thin layer, was moved from in contact with the ball lens to further and further away. So that gives you an idea of the depth sensitivity of the shallow channel of the probe. The dots here show our experimental data, and the solid line shows our Monte Carlo simulation, and we're within about 10% of the measured results when you compare our predictions. So what we did next then was to take that knowledge that we had measured about what's happening to the distribution of fluorophores throughout the epithelium and the stroma, and built that into our Monte Carlo simulation. So we could vary the thickness of those 5 layers, and we could vary the optical properties for each layer, the scattering and absorption coefficient, the scattering anisotropy, and the fluorescence emission. And then as our output, we could track what was the fluorescence emission spectrum, and we could keep track of those photons that were detected, what was the depth at which they were generated within the tissue. So we could determine, were we really confining our light to the epithelium with the shallow channel of the probe. These are the scattering and absorption coefficients that we used and the fluorescence emission properties that we used. I'm just going to skip through that and be happy to go back to it if there are questions. But I want to get to the results. So the first simulation that we did was we said, okay, if our shallow channel truly is collecting light only from the epithelium, and what I see with dysplasia is an increase in the amount of fluorescence from the basal layer, if I build that into my simulation, so here I have my baseline case for the normal tissue, and now I build in this increase of fluorescence in the basal epithelium, what would I predict should happen to the fluorescence? Well, in fact, what I predict is that the fluorescence intensity should go up, and that it should shift a little bit to longer emission wavelengths. Well, it's not what I measure. I do measure the shift, but you'll recall all of our data showed a decrease in the fluorescence intensity, not an increase. And if you look at the fluorescence photons that you detect, and you figure out how many of them were generated within the epithelium, in the baseline case, only about 40% of those photons that I detect are generated in the epithelium. More than half of them are actually generated in the stroma. And as I increase the basal epithelium, I get up to about 60% of the detected photons are coming from the epithelium. But even with this ball lens tipped probe, I'm still getting signal from the stroma. And so what we did next was we looked at what happens if you change the optical properties of the stroma. And we changed it in 2 ways. First, we changed just that superficial stroma, that 125 microns right beneath the basement membrane. And then we looked at changing the optical properties of the deeper stroma, because we wanted to understand how deep are we probing with our ball lens tipped probe. Okay, here's our baseline case now, And now what I've done is I've dropped the fluorescence efficiency of just that superficial layer of the stroma. And you can see as I do that, I go from the baseline case down to the prediction, which is much more consistent with what we actually measure. And you also see now the shift over to longer emission wavelengths. If I leave alone that superficial stroma and I only change the optical properties of the deep stroma, you can see there's very little effect. Here's the baseline case. Here's changing the optical properties of the deeper stroma. So with this particular probe geometry, we're sampling the epithelium and about the top 125 microns of the stroma. And when I look at the fraction for normal tissue, the fraction of total fluorescence that comes from the epithelium, it's about 43%. If I look at the fraction of the total fluorescence that comes from the epithelium and the top 125 microns of the stroma, about 90% of the light is coming from there. So these results are consistent with that. So that tells you sort of on average that we can use Monte Carlo to help us understand the results that we're measuring. But what it doesn't tell you is that for a particular patient and a particular site, if I measure the optical properties for that site, are my predictions of the Monte Carlo consistent with what I actually measure? And so we did a study where in patients, we measured the tissue in vivo from several sites. And here's the spectroscopy data from one patient. Here's a normal site, a dysplasia, and a cancer. These are individual sites from the same patient. And then we immediately biopsied those same sites, and we sliced them, and we imaged them using the Confocal fluorescence microscope. So here's the normal tissue, the mild dysplasia, and the cancer. And you see in the mild dysplasia, the increase in the epithelial fluorescence, the loss of the stromal fluorescence, and you see the disorganization of the cancer where you have tumor cells surrounded by the stromal matrix. And now if I use the geometry and the intensity from these measurements in the context of my Monte Carlo, I can compare my measured data, which is shown with the blue solid lines, to the results of my Monte Carlo prediction, which is shown by the red squares. And as I go from normal tissue to dysplasia to cancer, you see that I can predict both the drop in the fluorescence intensity and the shift to longer wavelengths. And so it turns out that if you track where these fluorescence photons are coming from, that in the normal tissue, we find that about half of the photons are coming from the epithelium. So the fraction of photons coming from the epithelium are shown with the black bars, and the photons coming from the stroma are shown with the open bars. So this is the normal tissue here, and now this is the dysplasia. And in the dysplasia, almost all of our light is coming from the epithelium. Only a bit of it is coming from the stroma. And so the reason that we see this drop in the fluorescence is that we're getting less of this light from the stroma, and the stroma, as you saw before, is much brighter than the epithelium. The shift to longer wavelengths is caused both by the increased amount of epithelial fluorescence that you detect, as well as an increase in the hemoglobin that contributes to a little bit of the reabsorption of light that comes from the stroma. So in the remainder of my talk this morning, what I'd like to do is talk about some new technologies that we're very interested in to be able to image in vivo with higher spatial resolution and to provide greater molecular sensitivity. And so I'm going to talk about doing microscopy in vivo, and then talk about developing contrast agents that we can paint on in vivo and use as a source of improving contrast in our in vivo microscopy. So, the traditional histopathology slides that we show you, we take the tissue out, we slice it, we stain it, we look at it under a microscope. So what we want to do is be able to get that kind of resolution in vivo and to develop the same kind of stains that give rise to this contrast in vitro, but be able to do that in vivo. And our approach there is to use in vivo confocal microscopy as a way of basically gating out the defocused photon. So if we place a pinhole at a conjugate image plane in our confocal microscope, light that's coming from our focal plane will pass right through the pinhole, whereas the out of focus light below and above this plane are mostly rejected by the placement of the confocal pinhole. And just to give you an idea of the kinds of images that you can acquire, this is an image of skin that was made using 785 nanometer illumination, and we're looking at the reflected confocal in this case. This image was obtained about 75 microns beneath the surface, here 125 microns beneath the surface, and here 200 microns beneath the surface. In skin, what you see are dark nuclei and bright cytoplasm because the melanin granules that are present in the cytoplasm give rise to bright reflectance. And basically our contrast in these images is provided by discontinuities in the refractive index. Here we can see the tips of capillary loops, and you can actually watch red blood cells flow through these capillaries because you can do this imaging at video rate. And then we can just get down into the stroma and see collagen fiber bundles in the stroma. Because there's melanin present in the skin, we see this particular pattern of contrast. When we look at an amelanotic epithelium, such as the oral cavity, instead what we see is that the nuclei appear bright and the cytoplasm appears dark. So I'm going to play a video, and initially in this video, the focal plane of our microscope will start at the surface of the tissue, and it will move deeper through the tissue. And so the white dots that you see are nuclei, and you can see that as we move deeper, the nuclei are going to become larger and become more closely spaced as we get down here to the basement membrane. And I can play that one more time. So here we're at the surface. We've used acetic acid, just a vinegar solution, as a way to enhance the refractive index mismatch between the nucleus and the surrounding cytoplasm. And with this technique, using these near-infrared wavelengths, we're able to penetrate between 300 and 400 microns, which is typically enough to see through the entire epithelial thickness. Now, this image was made with a non-fiber-optic confocal microscope, and you can use it to image tissues in vitro or to image sites like the skin, but it's difficult to image the oral cavity or any tissue where you would need endoscopic access. And so our solution to that is to use a coherent fiber optic bundle and basically to raster scan our beam across the back end of this fiber bundle so that in effect each fiber within the bundle acts as a confocal pinhole. The individual fibers in our bundle are typically 4 microns in diameter, and then we demagnify that so that our spatial resolution is about 2 to 3 microns with this device. But one of the challenges of imaging through a bundle like that is by the very nature of that approach, your resolution is not limited by diffraction. It's now limited by the Nyquist sampling because you're sampling through that fiber bundle. And you can see the pixelated nature of the image where you can appreciate the individual fibers within the bundle. Nonetheless, you can still see the nuclei. This is an image that was obtained of the cervix. This is a normal image on and a dysplasia on the right, and you can appreciate the increase in the nuclear size and the nuclear density. This is the probe that we built to measure this type of image in vivo. And here you can see an image that was obtained from the oral mucosa, starting at the surface and moving deeper into the epithelium. And again, you can pick out the individual nuclei that were obtained with the fiber bundle. We've also done work to look at, can you analyze these images to automatically recognize whether or not you have a high-grade dysplasia? And to do that, we basically segment the images to identify the nuclei and then measure features like the nuclear size and the nuclear to cytoplasmic ratio. And what we found is that the nuclear to cytoplasmic ratio gives you a much better indicator than the nuclear area does. This was from a pilot study looking at cervical biopsies, and the black dots are high-grade dysplasia, green is normal, and the red is low-grade dysplasia. And you can see we get very nice separation using a fairly simple feature. More recently, we've developed a much simpler alternative to the fiber optic confocal microscope. It's a system we call our needle biopsy system. And this again uses a coherent fiber optic bundle that's just placed directly in contact with the tissue. And then essentially you have nothing more than a standard fluorescence microscope here. You have your LED light source, a dichroic mirror, and then a tube lens which focuses your image onto a CCD camera. Here's the fiber bundle that we're using. It has an outer diameter of 900 microns, and we're able to achieve a spatial resolution between 3 and 4 microns with this device. And we've been testing this device in fluorescence mode using a contrast agent called acroflavin. Acroflavin binds nonspecifically to DNA, and so it makes the nuclei brightly fluorescent. And so in collaboration with Anne, here's a resected tumor from the oral cavity. We've done a topical application of acroflavin. The yellow line shows the clinical margin of the lesion, and here is the needle biopsy probe. And we're going to obtain an image here from a normal area and here from an area that was in the tumor. Here's the image from the normal area. You can see the bright nuclei. They're fairly small and far apart. In contrast, in the area from the tumor, you see the nuclei are much larger and more crowded. And if we extract out the nuclear to cytoplasmic ratio, before we found that the line dividing the dysplasia from normal was at 0.08. So I've just drawn that same line here. And you can see that using that same discriminant, we get a very nice separation between our abnormal and our normal. We've used this in normal volunteers. This actually is me that you're going to see a video of from the oral cavity with topical acroflavin. And here we're just moving the fiber probe across the oral mucosa, and you can appreciate the nuclei that show up in the image. The images that I showed you previously were obtained with a non-scientific grade CCD camera. We put together the total system for about $2,500. If you use a scientific grade CCD camera with a higher dynamic range, you can improve the image quality. So this system, again, the total cost was $11,000. So relative to a confocal microscope, we're still talking about a fairly inexpensive type of system. So finally, we're interested in contrast agents that will allow us to look at some of the molecular changes that are of interest clinically. And we've been looking at a variety of different types of contrast agents, ranging from small molecules to peptide-targeted contrast agents, to antibody-targeted contrast agents, and then even looking at nanoparticles that can be targeted with various targeting moieties. And in each case in our contrast agent, there are two components. There's one component that's going to bind to the biomarker of interest, and another component that will provide the optical signal so that we have something to detect. And I thought I would show you a few results from some pilot studies that we've recently completed. The first contrast agent I'm going to show you is an example of a small molecule. This is a deoxyglucose that has been labeled with a fluorescent dye. So the contrast agent is called 2-NBDG. It consists of the deoxyglucose and the fluorophore. And what happens when you apply this deoxyglucose is that cells that have glucose transporters will take this up. It becomes phosphorylated then and trapped within the cells. And because tumor cells overexpress glucose transporters, they will uptake more of the deoxyglucose. And this is the basis for FDG PET. And here, instead of putting a radioisotope on the deoxyglucose, we're putting a fluorophore. So what we did was we applied this topically to an abnormal oral biopsy, and then we sliced the oral biopsy and we looked at it under the fluorescence confocal microscope. Here is the epithelium, and here is the stroma. And what you see is that here the entire dysplastic epithelium is taking up the contrast agent, and you see the dark nuclei because this contrast agent is localized to the cytoplasm. So here's the dark nuclei with the cytoplasmic uptake of the fluorescently labeled deoxyglucose. If I compare the uptake that I get in a normal oral biopsy to that of an abnormal oral biopsy, you can see substantially more uptake of the contrast agent. And these were taken with the same laser power and the same gain. In a study that looked at 16 biopsies, they were paired biopsies obtained from the same patient. We obtained wide field fluorescence images before we applied the deoxyglucose, and then applied the deoxyglucose and obtained wide field images after that. And we looked at the increase in fluorescence post relative to pre. And what that allows us to get at is two things. We know that our abnormal tissues have decreased autofluorescence, so they start out lower. But we know that they take up more glucose, so they end up higher. And so by combining those two features, what we're able to get is a very, very high differential contrast. So this is the differential contrast. This was a biopsy pair that was cancer and normal. You can see the differential contrast is more than 30. So here are all the cancer normal pairs, and we have differential contrast ranging from 25 up to 40. When we look at cancer and mild dysplasia, we have differential contrast of about 20, and then this was a moderate dysplasia relative to normal. So by combining those two features, we're able to really maximize the amount of contrast that is available. Tumor cells, many of them overexpress a surface receptor called the epidermal growth factor receptor. It binds a ligand called the epidermal growth factor, EGF. So we can take EGF and we can attach a fluorescent dye to EGF. And this is an example of something that has a higher molecular weight than the deoxyglucose, but it's much smaller than an antibody, for example. And so we've looked at what happens when we label normal oral mucosa and abnormal oral mucosa with this EGF targeted fluorophore. And you can see much greater uptake in the abnormal tissue. And the pattern of staining now is right around the cytoplasmic membrane, which is where that receptor is expressed. And now if we look at the differential contrast, again, we have 8 pairs of biopsies. Again, we imaged pre and post the EGF. Our dye in this case was Alexafluor 647. And so we're exciting around 650 and we're exciting around 630 and looking at the fluorescence around 650. In this case, there's very little difference in the autofluorescence, and so all of our contrast is coming because of the increased uptake of the dye. And you can see it's not as high. It ranges from 2 to 7 in this case. So taking advantage of that autofluorescence, it can increase your differential contrast significantly. And then finally, we've been looking at metallic nanoparticles, such as gold and silver nanoparticles. These are the largest of our contrast agents, ranging in diameter from about 20 to about 90 nanometers is what we've looked at. And we can target these with things like nucleic acids, with peptides, or with antibodies. And what I show here is, this is a cervical biopsy that we've labeled with gold nanoparticles that are conjugated to an antibody that binds the epidermal growth factor receptor. And we conjugated to this antibody both a metal nanoparticle as well as a fluorescent dye. And you can see the cytoplasmic membrane labeling both in reflectance mode and in fluorescence mode. So what's the advantage of having the metallic nanoparticle? Well, the advantage is that when these metal nanoparticles aggregate together, you see an enhancement and a shift in the surface plasmon resonance, and that's what we're essentially sensitive to when we image these in scattering mode. So these are CIHA cervical cancer cells that have been labeled with those antibody-targeted gold nanoparticles, and we're looking at them in dark field microscopy. And so you can see in the low-expressing cells that don't have very much EGFR, we see the green backscattering associated with single gold nanoparticles. But the high-expressing cells, where we get the aggregation of these nanoparticles, they're bright orange, and when we measure the spectrum in dark field, you see both an enhancement and a shift of that surface plasmon resonance. What that allows you to do is to translate roughly a 2 to 3-fold increase in your receptor concentration into a 10 to 20-fold increase in the signal that you measure. And so if we look at, again, pairs of biopsies, here we're looking at cervical biopsies where we have a benign condition, HPV-associated changes, and then increasing grades of dysplasia. If we look at the signal of the abnormal divided by the normal, it goes from about 1 to about 20 as we increase from the benign changes to the dysplasia. And there's only about a 3-fold increase in the concentration of the EGF receptor. But by using the localization and the aggregation of those gold nanoparticles, we can turn that into more contrast in our image. So in summary, what we're interested in doing with some of these new techniques is making a transition from the current clinical paradigm where we can use in vitro cytology and in vitro histology to look at these changes in the cellular morphology and the tissue architecture, but instead be able to do that in vivo using confocal microscopy and to automate the interpretation of that. And instead, now we can only look at these molecular changes in vitro using things like immunohistochemistry or PCR. Instead, we'd like to be able to look at that in vivo using targeted contrast agents. So let me stop there. I'd be happy to take any questions.
Questions?
Well, very nice, the explanation of the fluorescence. I'm going to start from the end concerning the nanoparticles. Well, this is a more general concern. Life is not used to deal with nano objects. And my question is, if we're going to start to use nanoparticles in clinical uses, there is any consequences that we may have in the future? Like creating, modifying reproduction, because they also the particles will also attach to DNA and all that, right?
So one of the advantages of using gold is that gold is fairly inert. It's also fairly biocompatible. In fact, people inject colloidal gold to treat arthritis at much higher concentrations than we are talking about using at 3 orders of magnitude higher concentration than would be necessary to do this kind of labeling. We have done some preliminary cytotoxicity assays looking at cells in culture that have been labeled and not labeled, and we're not able to detect any cytotoxicity, both for short-term exposure and for exposure on the order of a week to 10 days. And so I'm encouraged by that preliminary data. I mean, clearly there will need to be more in-depth studies in animal models to assess the toxicity. But I think given the amount of clinical experience already using this type of particle, I'm not so worried. I think when you talk about using quantum dots and some of the semiconductor nanoparticles, I think there it's actually much more of an open question as to whether there will be some serious concerns about the safety.
Oh, the question is more exploratory. How many generations will take for us to identify if we really have changed individuals by putting these nano objects inside? No, I'm a fan of using nanotechnology.
I mean, I think
I'm just trying
I think
to see if you have thought about this kind of thing.
No, I think long-term animal studies are really one of the best ways to address that and to look at the clearance mechanisms. I'm less concerned for topical applications because you're controlling where you're going to put these nanoparticles, and if we're able to show that they give specific binding to tumor cells, typically though you're going to remove that tissue. So I think it's sort of the safest way I can imagine introducing.
I think I miss Rebecca. How did you apply the nanoparticles?
Topically.
Topically. And they reach everywhere, right?
So,
but you show that they aggregates in the...
Yeah, so it's a really important question in terms of how much tissue penetration are you going to get. And we have in our lab conflicting data on this point. So we have shown consistently, both in cervical tissue and in oral tissue, that for contrast agents that are smaller than about 10 kilodaltons, we get complete penetration of the epithelium. And so for things like the Alexa Fluor targeted EGF and the NBDG and the acroflavin, you get essentially instantaneous and complete penetration of the whole epithelium. And even in tumors, we can see penetration down as deep as a couple of millimeters with things that are that small. with the gold nanoparticles, what we find is that about a third of the time we get penetration throughout the abnormal epithelium, and we don't get much penetration in the normal epithelium. We're looking at permeation enhancers that we can use in order to be able to get better penetration in the dysplastic epithelium. And so it's an area of very active study in our lab right now to look at the trade-off in terms of the size of the nanoparticle, the size of the targeting moiety, and the use of permeation enhancers. So we're looking at things like transcutol and other substances that people who are interested in gene therapy are using to get permeation of their liposomes for topical permeation.
Any more questions? So I guess we're gonna thank you again for the both nice talks.